World's greatest. I am a mountain, I am a tall tree, oh, I am a swift wind, sweeping the country. I am a river, down in the valley,
We first arrived here, I remember it was on the 20th of August 2010, and my son was just 5 years old now, then. And obviously we didn't know anybody, but the whole idea of coming here was to pursue a dream of getting that PhD. Mm-hmm. The first place, before I came here, I already knew I was coming to. That was to work with Dr. Brian Bothner, and it's been a pleasure. So the project that I have been working on in collaboration with Dr. Walid Mati, who has since left the lab in 2013. And with him, we've been working on heavy metal stress, on extremophiles, and to be more specific, on sulfolobus sulfataricus. So my main interest was to see how this extremophile behaves or adapts to very high concentrations of heavy metals, and the two heavy metals of interest has been antimony and arsenic. So I wanted to look at any protein regulation upon stressing this microorganism with heavy metal. And okay, now being 2014, I'm very excited that I was able to complete my studies, and I was able to do it under 4 years. So to me, it's a very great achievement, and it's also an indication that I had a very good mentor, or should I say mentors, which are Dr. Walid, Dr. Brian Bothner, and Dr. Markia, who actually in a special way pushed me to reach the destination of getting this PhD. If there's one person that I really wish was here to experience the gratitude that is within me, it's Dr. Mati. But one of these days, I'm hoping that he will really get the message across on the impact that he made in my life, and not only him, but his family as well, because you see, we are both African brothers, and me being a single mother, it was very difficult to juggle everything all at once. And him, his wife, and his kids, they came through to actually help out unselfishly, free babysitting, actually, if I have to say that. But it's just been a pleasure and a wonder that I was able to come across Walid and the Bothner Lab. They've been somewhat supportive as well, but most notably Ravi has always been available. Dr. Monica as well, and of late the gentleman from Saudi Arabia that doesn't have oil in his backyard, with his electronic skills and his willingness to always be willing to help out and carry out experiments without any complaints as well. But also, it would be a very big mistake for me not to thank the rest of my committee members. A lot of people always complain about how it is difficult to pull all of them together, but in my case, that has never happened. I was not able to experience that. Last but not least, the Lord God Almighty, and for the special one here, my son Vui. Honestly speaking, we should be sharing this thing together, because he's been there with me from the beginning up until now. Yeah. And if anybody that did PhD, they will know that it comes with emotions that cannot be described, and this poor guy has absorbed all of them. So and hopefully one day he will realize that everything has been done so that he can get a very brighter future that I did, and take a less turbulent pathway that I, as a black South African, had to take.
Patricia, right? So what I can remember about Patricia is that we met, we talked first time in 2011, when I was doing the TA, for my first semester, I met Patricia. And that time, we did not communicate much, but that was the first glimpse I saw about her. But along with that time, I started knowing Patricia more and more. And then I realized that she's the person, who can be a really good friend, who is, very warm when you meet the person. She always asking about you, your families, and, about the work and the progress. So she's a very, very comfortable person to talk. So I can share my problems with Patricia a lot. And she's a wonderful friend. Well, I would say if you talk to anyone about Patricia, you will find that she's the person, anyone want to be a friend. She's very generous, very social, she has maintained everything, beside that she was a single mother for a long time, and, she maintained her, personal life and the professional life really well. She finished her PhD in a decent amount of time, 4 years being a single mother. I think this is, the commendable job that she has done, and there's certain people, they should be looking at her as an example, Well, so, if you ask me about Patricia, I would be more biased because I'm a good friend of her. But, but from a neutral point of view, I see Patricia, she is a very meticulous, a very precise and very direct kind of person. She's very expressive. You can see and know about Patricia from her face because she's very expressive as well. I've seen her working, the very odd hours during the days and sometime on the weekends, in order just to accomplish something, in a given time. So she's a wonderful person. She's a very good scientist. She's a wonderful mother. I wish I'm going to get these kind of friends always in my life. So I'm going to give the best wishes to Patricia for a wonderful life. It's okay.
So you'd like me to say one or two things about Patricia Mathabe, right?
Yep.
I came to Bozeman in 2011, August 2011, as a Fulbright. And I got to hear that there's another Fulbright from South Africa. And naturally, because South Africa and Zambia, they are in the southern part of Africa, I got to connect with her. So I've known her since that time, and together I found that she was trying to set up the African Society at Montana State. And that's how I also got interested in that society at Montana State University. And then she impressed me with one thing. Being a single mother, she had a son she was looking after, at the same time pursuing a PhD, not in any easy subject. She was pursuing a PhD in biochemistry. So you can imagine she had to run between errands of taking the boy to school, fetching him back, preparing his meals. At the same time, she had to run some science-based investigations at the laboratory. I remember on several occasions she would say, "Look, I need to run to the lab. Could you please remain with my son for just 15 minutes? And I would sympathize and go and kind of babysit Vui. He was already a big boy, in order to allow her to go and attend to her experiments in the lab. So she is a lady who I can say is self-motivated, resilient, and she doesn't give up that easily. And I would like to wish her success as she embarks on this new life of being a graduate. She defended successfully. We all attended her defense, and she's preparing right now to go into another phase of her life. We wish her the best. Thank you.
So it's a real pleasure for me to have Patricia at this point where we're ready for more dissertation defense. So just a little background about Patricia. So as you all are aware, she is a native from South Africa. She got her bachelor's degree in biotechnology from Ball University in 2005, and then she went to Cambridge University and received her master's degree there. Along the way, she has received a number of awards, including a Nelson Mandela scholarship and a Fulbright scholarship, which allowed her to come to MSU. And I can still remember receiving an email about Patricia and the potential for, starting in this whole process, which now we're right near the culmination of, and thinking, wow, this is really interesting. And so it's amazing just from, this initial email contact and then talking with Patricia back and forth, for a month or two to try to, is this a right fit? Is this a good place for you to come? Montana in the middle of nowhere. Are you sure you know what you're getting into? And so, she has come here and, done a really nice job that she's going to tell us today about her proteomics work and differential analysis of the effects of heavy metals on sulfolobus, everybody's favorite archaeal organism, at least in the room. I think it's our favorite. But a couple of other things about Patricia. One, I know she enjoys music very much, and I actually a couple of months ago, with the passing of Nelson Mandela, she was actually asked to kind of provide a little opening and background to the movie A Long Walk, which was shown at the Emerson Cultural Center. And so Patricia and Ilse Marie actually performed the South African national anthem. And Patricia sang the anthem while Ilse Marie played on her cello, and it was quite a moving experience for that. So she does more than just her scientific work. She's also a single mom, raising a wonderful boy. And so, Patricia, excited to hear about your project and work here.
Thank you, Brian. Thank you all for coming to listen to me talk about the work that I've been doing for the past 3 years, working towards my PhD. So the title of my talk is the proteomics and the in vivo labeling of protein thiols in sulfolobus sulfataricus during exposure to antimony and arsenic. So this is just an overview of my presentation. I will tell you about the objectives of the study and why I really got interested in working in this project. The heavy metals of interest, and then my talk is divided into two. The first part will be elucidating the differences in protein abundance post-treatment with arsenic and antimony. And the second part is about a method, a very cool method that I developed to label cells or proteins in vivo while cells are still intact. And like Brian said, I'll be working with I worked on our favorite organisms, which is Sulforubus sulfataricus. And the reason why is it's a very interesting organism in that it is found in very extreme environments or conditions. It is an archaea. It lives in environments with very high temperatures, which is between 70 and 90, acidic, 2 and 4. And also it has there's some heavy metals, some level of heavy metals that is found in the environments that they grow in. It is also found just next door from us, the Yellowstone National Park, and also worldwide in volcanic systems. And just because of all this, it makes it a very interesting, or it makes it a model organism for studying life in extreme environments. Now, just to be background about these metals of interest, which is arsenic and antimony. So they occur naturally in the Earth's crust, and in most cases, they are found to be coexisting together. They're found in different oxidation states, but predominantly in oxidation 3 and 5. They have been named to be a source of pollution, in which one common source of pollution being mining. and they're very toxic. So this is just to show you how the pollution of these heavy metals, or in this case it's just arsenic, how it causes a problem worldwide from North America, South America, Africa, and even Asia. Now, it does have some health impacts because they have some health impacts because of the toxicity, and in the same light, they have found good uses to humankind. Arsenic can cause cancer of the skin, liver, lungs, bladder, and kidney, and it has been reported to cause some birth defects. And then you have antimony, which causes cardiovascular diseases. It slows growth rates in infants, and there has been instances where it caused spontaneous abortions. And for both of these in high concentrations and maybe prolonged contact with these heavy metals, it results in death. Now, uses, arsenic is used for the production of as agents in insecticides, rat poisons, and for those who need to tan, luckily not me, tanning agents. And antimony, it's found in trace amounts in antiretrovirals, which are used for the treatment of HIV, and in drugs used for to treat tropical diseases like leishmaniasis. And additionally, antimony has been it's been used to help dissolve other metals. Because they're found in the environment, the obvious thing would be they do interact with microorganisms and, of course, plants. Now, microorganisms have evolved or have come up with systems or homeostatic mechanisms which help them to deal with these heavy metals. For example, in eukaryotes, our yeast, Saccharomyces cerevisiae, uses phosphate transporters to transport arsenic 5 into the cell. And then it also has a glycerol facilitator, FPS1, which is an aquaglycerylforin, which normally transports water and glycerin into the cell. And so it helps with the transports of this inside, of arsenic inside, and also helps to extrude it out of the cell. And then you have prokaryotes. It uses the GLPF gene, which transports the arsenic inside and then outside as well. And then in archaea, there's this arsenic-resistant ARS operans, which encodes genes that extrudes arsenic 3. Now, antimony, same thing. Now, I need to mention this, that there is a plethora of data or research that has been conducted on the interactions of microorganisms and arsenic, but then data about antimony is just not as much. So most of the information that we know is normally is in most cases in reference to arsenic, because it is believed they both have they both belong to group 15, and they could have, or they have similar chemical characteristics. So just taking one, prokaryotes, E. coli, transporters RSB and ACR family are used to mediate arsenic, and it also mediates antimony 3 efflux. And then again you have the glycerol transporter gene GLPF1, which reduces the uptake of antimony 3. Now, one of the other issues or the problems that these heavy metals cause, or the effect that it has on microorganisms, is that in its presence, it causes oxidation. So it causes, which leads to oxidative stress. So during oxidative stress, what happens is there is an imbalance between free radical generation and the antioxidant defense system. It induces reactive oxygen species, which produce free radicals, leading to the damage of biomolecules. Then what also happens is that the thiol function groups of the amino acid cysteine can experience a broad array of oxidative modifications, and and performs a number of physiological functions. Now, why did I go into this research? So the whole idea was to to gain insight on how antimony and arsenic, what effects they have on the proteome of sulfur lobus, sulfur ptericus. And I also wanted to come up with a method that would actually help us to try to get a true representation, rather, of what is happening on a level of thiols. And then I also wanted to understand the level of oxidation of sulfur lobus, sulfur ptericus proteins by antimony. Now, this is just a schematic view of how I started my project. Now, the first thing that I had to put in mind is, I am trying to look at the proteins, or of the changes of the proteome, and ultimately genes. I want to find out what is actually happening when antimony stresses sulfur lobus. So I was not interested in the dead cell. So I needed to choose the concentration of the heavy metal, which will slow the growth, but then toxic enough to give us an idea of what is really happening while the growth is slowing, the growth rate is slowing. So now what I did was I had to culture my sulfur lobus cells in an environment which mimics the environment that they grow in, but obviously it cannot be entirely. So I grew my cells at 80 degrees Celsius, which is quite ideal for our sulfur lobus to grow. And then when it reaches a certain OD, I stressed my cells separately with antimony 3 and arsenic 3. And then because I wanted to find out the relevant, or the ideal concentration of my heavy metals, I tried out 3 different concentrations: 50, 240, and 1,000 micromolars over a period of time. And then I collected or harvested my cells after about 8 hours. And then started with my protein abundant project, which is 2D Dige. Now this is to those, some who didn't, because we work with it a lot. So this is just to give you a brief explanation of what Dige is. So it is differential gel electrophoresis, and we normally use a family of cydies. So this method actually is very cool and ideal for us for 2 reasons. One, this process of 2D gels, or Dige normally of 2D gels, is very long and it's very time consuming. So when these came onto the market, what you do actually is you use it to compare 2 different samples, one stressed and one unstressed, diseased or healthy one, undiseased. So what you do is you take these chemical probes, you mix them with your samples, and then because what the chemical probes themselves, they have different tags which fluoresces at different wavelengths. So you mix them, and then after mixing these 2 samples, you can run them on one gel instead of running each sample on different gels. So it also makes the comparison or the analysis of differential expression to be to have less variation, because you don't have to deal with gel to gel variation. So this is the workflow of the whole part 1 of my project. So this is after 2D running the 2D gel, we scan them, and then we compare the stressed between stressed and unstressed. In this case, I had stressed, and then I had two samples which were stressed with different concentrations, which is 50 and 240. I overlap them, and then from there I look at any protein differential expression between the stressed and the unstressed. And then once I see proteins of interest, we excise them, we cut them out, and then we try to trypsinize them with using a protease of interest. So in this case, we use trypsin, and from there we take the peptide library, send it to mass spec during MSMS, and then we get we get a peptide sequence that we can then take it to a search engine called Mascot. So Mascot is a search engine that is connected to different databases, which has information on the peptides of these peptide sequences. Then it comes out and tells me, look, using this sequence, this is your potential protein of interest. So now, getting back to the results. Remember at the beginning I told you that I had to choose the ideal concentration of my heavy metals. So in this graph here, you're looking at sulfolobus response to antimony 3. It's a growth curve. And in this one is a response to arsenic 3. So on your y-axis, you're looking at that's the optical density. And then on the x-axis, it's the time that we collected different fractions, or that we observed the growth pattern of sulfolobus in the presence of these heavy metals. So now what we were able to take out of this is this. Indeed, these heavy metals do slow the growth of sulfolobus. And then we found that we ultimately chose this concentration, 240 micromolar, because indeed it does slow the growth of our organism. But at the end of the day, I think we thought it would be potent enough, but then it wouldn't kill it. So I would be able to, to do my studies. And also comparing these two, if you look at 240 with arsenic, the growth rate is actually, it's quite comparable. So now I then went to do DIG, and this is just a gel representation of all the protein spots that I compared between stressed and unstressed. And for this experiment, I found out that 9 different proteins were actually significantly, they changed in abundance significantly. And then the next thing was to, so now this is just a blow-up to show you how I actually choose, or how the software tells me which of the proteins are significantly changed or regulated. So this here is the untreated, this panel. It comes from the untreated gel in samples, and this is treated. So this is the same protein spot across the treated and untreated. And then just by the look of the eye, you can see that in this spot which comes from the proteins that have not been treated, and this is from treated. So you can see that the intensity increases, and then that's when the software that we use tells us that, hey, there's a change in abundance there. So the next thing was to identify those proteins, and out of those 9, I was able to identify 5 of those. And this is the molecular weight, the fold difference, and the peptides. And the p-values is less than 0.05, which is 5%. And now all of these proteins that I identified are actually stress-related proteins. So one is prefoldin, which is a molecular chaperone found in eukaryotes and in archaea domains. So what it does is it participates in the refolding of the misfolded proteins that could have been caused, or that are caused by our antimony when we stress the cells. And then prefolding, it's one of the proteins that were upregulated, then comes in and try to to fold the protein again. And then you have your carbon monoxide dehydrogenase, which is associated with your ribofurans. So ribofurans play a role in the detoxification of ROS by reducing hydrogen peroxide. Then you have some like ribonucleoproteins, which are a large family of proteins. Which are involved in RNA metabolism, and then inorganic pyrophosphatase, which catalyzes the cleavage of pyrophosphate into 2 phosphate molecules. So I repeated the same experiment, but in this time I used the cells that were stressed with arsenic-3. So, but then what was interesting is with this, instead of I was able to find 29 regulated proteins. So again, I did those proteins, and there was quite a number of stress-related proteins. You have your DNA binding protein, which is a transcriptional factor, or that is normally that has been identified to take part in. It's also a stress. Protein or stress-related protein, and then the others as well. There's quite about 50% of those. Now, what boggled my mind was, okay, fine. So these are the 2 metals, sulfur lobas and antimony. Arsenic and antimony. So with antimony, I was only able to identify 9, and then with arsenic, I identified 29. So the first thing to do is to look, wow, is it a human error? Did I do anything wrong? So 2 things that I did. The first one was to repeat it because Brian said so, and then I repeated the whole process, and I got more or less the same results. Less proteins were regulated. And then that's when I said, okay, fine, I repeated it again. What do I do next? So the next thing was, okay, fine, let me try to just see if the quality of my gels, what the quality is or is like. So what I did was I plotted this, it's a volcano graph, which on this axis, that's the fold difference amongst all the proteins, and this side is the downregulated proteins. And here, this line here, it's unregulated proteins. So what I did is of course in these axes, these are all the these lines, different protein spots that I used to come up with this graph. So, and then I calculated the coefficients of variation. Now, what I found is my coefficient of variation is less than 1%, and normally anything that is less than 3% is considered good. So that mine was less than 1%. It just shows that there was really nothing wrong with my gel, so there's something going on. The question is what? So, but then anyway, just to give you the the summary of my findings was I was able to determine that the presence of both antimony-3 and arsenic-3 slows the growth of sulfur lobus sulfataricus, and there's a specific set of proteins that were regulated. 29 proteins were regulated when cells were treated with arsenic-3, as opposed to the 5 that were regulated after treatment with antimony-3. The coefficient of variation was less than 1% between biological replicates, and then all the regulated proteins were cytosolic. So then, which got me thinking and said, okay, fine, where do I go to from now? Then some of the things that came to my head was like, wow, wait a minute, I was working with cytosolic proteins. Could it be that some of these proteins that are regulated are in the membrane proteins? And which in many cases, it's very, the 2D gels, you know, the membrane proteins, they're poorly represented there. So, And then that's when I said, okay, fine, maybe I should try to zoom into the membrane proteins to find out what's going on there. And at the same time, try to look at, I wanted to look at the free thiols, the changes of the free thiols post-dressing with antimony. So now, that this is actually the second part of my project, moving away or moving from the 2G gels. So in this method, we actually used these cool melamine reactive probes, which are synthesized by Ben Reeves in Paul Greco group. So this is just how these tags look like that I'll use for this this next experience. So they normally label your cysteines or free thiols, and what you're looking at here you've got your tag. This is actually what will fluoresce when you scan your gels, and then you've got this linker, and this is the reactive group. So now this, what's cool about this, the melamine reactive probes that we have here at MSU, or that Ben synthesizes, that they have this very cool zootyanic structure, which helps it to be more soluble as compared to what's available what's available out there. And and they're very robust as well. So that's when I said, okay, fine, let me use it. I used them to come up with the in vivo labeling method, which really I think it's the next cool thing after the creation of bread. So here, so what's cool about them is with this method is I can actually label intact proteins without breaking them, and at the same time I am able to label cytosolic proteins and your membrane proteins at the same time. And one nice thing is I was able to label sulfur lobas at very high temperatures because of the robustness of these chemical probes that we have. So now this is the gel that I ran from these fractions. So what you're looking at here is, in this gel here, I just used this sulfur lobas without any stress because I was just developing this method. So on this lane here, I've got a control. So I normally you have the control to see if after labeling this, I would spin down my sample, and then the supernatant that remains, I run it here. So this was an indication to tell me if I broke my cell while labeling, so that it is a bit quite clear. It shows that, you know what, I did label intact proteins. And here you're looking at your cytosolic proteins that were isolated after using the freeze therm method. And this is before I break my cells and make sure that I quench the reaction. So I know that when I'm breaking my cell, the reaction is not, it's not proceeding or continuing. And here you've got your membrane proteins, and then you've got your integral membrane proteins. And clearly, just by looking at the patterns of these different fractions, I would say that we are enriching, because you look at the differential expression between these two. And then this at the bottom is just the protein gel. It's the same gel that I stained in colorimetric Kumasi gel, and here you're looking at proteins, whereas here I labeled the thiols. Now, now that I was certain that I worked, that my method seems to be working and quite reproducible, I must say, the next thing was, you know what, I've been using this these probes that were synthesized here. Now, how do they differ from what is available out there commercially? So now on this panel here, these are the dyes, the commercial dyes, and in green, the in-house dyes that we have. So I wanted to look if there's anything special about, right? If there's any changes in, the labeling or the pattern, and clearly, it's actually quite comparable right? So then I was, say, okay, fine. But then the only difference is with these ones, you cannot run 2D gels on them. You cannot use them on 2D gels because they will precipitate, because they don't have that zootoionic molecule that we have in our labels. So then after that, that's when I started applying this method to my actual project, my actual experiments. So what you're looking here is these are the thiol labeled gel in which we labeled the thiols. So this is the molecular weight. And in this, the first lane, that's the proteins from the controls. They have not been stressed. And the second one, they've been stressed with 50 micromolar of antimony, and then the third, 240 micromolar of antimony. And over here, these are just peaks. It's like, it's the trace, the band is now, for example, in the control, which is black, it's comparable. It's actually, you're looking at these bands, but only displayed as peaks using the image J. So now, just looking closely, already you can see that there's changes across on the treated and untreated. That's the changes in protein banding. So in this one, this band over here, it's very faint, or it looks like as if it is not there. Whereas as we move to 50 micromolar and 240, there is an upregulation there. And then similarly, with this protein band over here, it's very quite intense. However, when you move across, there is nothing there. So clearly, there is something that is happening there. So now this is just to show you that everything, I ran about 36 different gels of these because everything had to be in triplicates. So this is just show you, and to show you that this method is actually reproducible. All the results are actually, so this is a triplicate of the untreated, 50 and 240. And this is just a fraction of the integral membranes, of the membrane proteins, and this is of the integral membrane proteins. And like I showed earlier, this is just showing the pattern of the bands, and you're looking at the intensity of the tag when it reacts or when it binds to thiols. So I identified some of those protein banding of the proteins that shows that were differentially that were upregulated or downregulated, and after treatment with your antimony. So these are some of the proteins. These are the proteins that I was able to identify. Now, notable ones. You have your temperature factor 55, which is your heat shock proteins, and it normally works in tandem with your pre-folding, and it helps with the refolding or the reformation of your proteins. Then you have your alpha monosidase, class C alpha monosidase, and in sulfolobus, it was found to take part in the processing of extracellular polymetric substances, and because these EPSs may they work in the processing of end lichens of proteins that are located in the S layer of sulfolobus. Now, the conclusion that I was able to come up with is that there is a regulation of some proteins which are stressed. There's a difference between how sulfolobus responds to arsenic and antimony. And then we were also, just by looking at the gels, we were able to see that membrane proteins have changes in thiol exudation states. And then the overall proteome has more, and then, so we were able to say that the proteome actually has, it's been oxidized after the exposure of antimony and arsenic. Now, that's the summary of the part 2 of my work. And this work, just to thank different people, my supervisor, Brian, for the patients, Mark Young made this really happen, my committee being very patient and being available all the time, and my mentor, all of the above that these people did. He played a very, very huge role. And then I've got, then there's Monica, Ravi, and Mohammed. These are my dogs that are my back. And then there's the Bosnia Lab, the rest of the Bosnia Lab, and the facility collaborators. Ben Reeves, he was all about the dyes. Thank you for making them available. And of course, Mark, who worked with him. And then made it possible, Fulbright, NRF, and Brian. I don't know which grant you used to fund me, but he funded me as well. So thank you.
I'm sure Patricia would be happy to answer some questions. Anybody has? I'll save mine.
Same here.
So when you label these at 70 degrees, the soft lobus, what pH and was it the low pH and?
I used, so what I did not mention is with these, it was at pH of approximately 6.8 because these the probes that's they label at pH.
Do you think that's an issue for membrane proteins that are seeing an acidic environment?
Well, it could be. It could be an issue, and that actually that state corresponds to. It could be the reason that when I was identifying some of these proteins, I couldn't identify a single membrane protein. So it could well be.
So you looked at the, can you go to the gel image that you had of the, yep, perfect.
This one?
Yeah, so it looks like when you're staining with the maleimide that you're getting less staining on the inside of the organism and more staining in the suggested membrane and integral membrane. Would that be a correct assumption?
Oh, well, yes.
Based on the fluorescence?
Yes, it does. It does look like, yeah, I did get a bit of, it's less intense indeed there than this. And yeah, that was actually what I saw across. It could be that maybe when I was breaking my cell, not much, I couldn't recover much.
Or it means the inside of the organism is
It's more oxidized than the outside.
Yep. That's a very cool result.
Thank you.
Okay. Any more questions? All right, let's thank Patricia again.